Hey guys, so recently we have been talking about prokaryotes and eukaryotes, so basically distinguishing between the two. Now we are going to take our focus and focus mainly on eukaryotic cells. Remember, prokaryotes are bacteria and archaea. They are very small, they do not have a nucleus, all of those distinguishing features that make them prokaryotes. Doesn't make them a weaker type of cell, it just means they're different. Another thing that I want you to understand and get a grasp on is the fact that we are talking about prokaryotic cells. So anything that's considered a prokaryote has prokaryotic cells. So I wanted to make sure that was clear. Now we're going to focus on eukaryotic cells and get into more details on the structures that make a eukaryotic cell. Some of this stuff will still cover prokaryotes, and you remember some of those common similarities between the two cell types. But again, we are focusing on organelles and what those structures do and what they look like. For the sake of time, because I do anticipate this video will be a little bit longer, I'm just going to go ahead and skip over the objectives and kind of talk about those in class. Here are all of the structures found in a prokaryotic cell, a eukaryotic cell, and some of these structures are found in a prokaryotic cell. So what I want you to do, if you can do this right now, hope that's great, and if you can't, that's okay, we'll work on that. I want you to identify which of these structures are found in prokaryotic and eukaryotic cells, and then decide which ones do not belong or are not a part of prokaryotic cells. That will really help you distinguish some of the differences when it comes to those two types of cells. We are going to go into all of these details in my notes. I do have 2 pages of notes, so please make sure you are writing and maybe leaving a little bit of space on the side so that you can add to those notes if you need to. Another thing that I really want you to realize and remember is that all cells in our body perform same basic functions, but they differ based on their location. So for example, talking about myself as a human, so I have eukaryotic cells. All of the cells in my body have the same DNA. They have the same function. So every single cell needs to get energy. It needs to respond to the environment. It also reproduces, so cell division. These are all things that every single cell in your body does. Now, obviously, there are some exceptions to those, and we'll talk about those, especially if you take human anatomy. But for right now, I do want you to know that every single cell in my body does the same basic life functions, and this is what keeps me alive. Now, what distinguishes the job of that particular type of cell, so for example, the difference between muscle cell and bone cell would be their location and where they're found in my body. So their job may be a little different, but they still all do the same basic life functions to keep my body going. All right, let's go ahead and start with the structures of the overall cell structures. And again, we are going to distinguish what is found in a prokaryotic cell and what is found in a eukaryotic cell. Plasma membrane is found in both prokaryotes and eukaryotes, and this forms a barrier between the cytoplasm inside the cell and the environment. This is something you should already know. We can think of the plasma membrane as like a security gate. Only things that are allowed to go in will go in. So you can think of, if you have, for example, I have a key card to come into the building. So if I use my key card, I have access and can walk through the door. Not everybody who has a key, not everybody can just come into our building. So we can think of the outside walls and the doors as our security system for the school, just like the plasma membrane is for the cell. It monitors anything that comes into and out of the cell. And how it does this, it's known as selective permeability. And what that means is that it allows only certain molecules in and out of the cell, which is a good thing. You don't want to just let your cells become influxed with water for no apparent reason. That could cause serious issues. Same with anything leaving. You don't want the cell to lose a lot of water either. That obviously causes other issues. So this is a really good check and balance of the cell, making sure everything that's in there should be in there. Everything that shouldn't be does not just flow through. The plasma membrane is really special, and the reason why it's so protective is because it's because it's composed of mainly phospholipids. So in the last chapter, when we talked about lipids, we talked about how those are like a fat and how water doesn't absorb through them, and I'm going to spend some time talking about that in class. The next thing that I want you to definitely know is that plasma membranes, depending on the organism, may have extensions on them, such as flagella and cilia. So basically, both of these things are used for locomotion or movement of some kind. Really, really helpful in getting from A to B, especially with single-celled organisms. These need some help to move. If they're doing everything in one cell, they can't just sit stagnant. They need to go somewhere. So flagella and cilia, and I'll draw a picture of what those two things look like as far as extensions come off of the phospholipid bilayer. The next thing we have is the cytoplasm and the cytoskeleton. Both of these are inside of the plasma membrane, so inside of the cell. It includes the watery gel-like material called cytosol. And remember, this is where all of the organelles are found in eukaryotic cells. In prokaryotic cells, in the cytoplasm, we have the DNA that's freely floating. We have the ribosomes, and obviously the cytoplasm. Big structure of this guy is that it helps give it weight, and it also helps give structure to the shape, it gets structure to the plasma membrane, which helps give it the shape that it has. So not all prokaryotes look like a circle. Some of them look like more of a cylinder of some sort. There's a lot of different shapes based on how much cytoplasm is present, how large the organism is going to get for a particular function. But basically, the function of the cytoplasm is just that structural support, holding things in place. In comparison to that, we have what's known as the cytoskeleton, just another reinforcement in a cell. Basically, it's like filaments or like threads that hold the shape of the cell. You can almost think of it as like a skeleton in our body. A skeleton in our body keeps the framework for our entire body, so we're not just like flopping around. Same idea with the cell. Now we're talking about the nucleus and the organelles. So from this slide on, we are no longer talking about prokaryotic cells. We are now focusing on eukaryotic cells. Remember, eukaryotic cells contain a nucleus and a lot more other organelles. They're very complex. We as humans are eukaryotes. Things like fungi, protists, plants, all of these things are considered eukaryotic cells. So there is much more greater diversity when it comes to eukaryotes. The nucleus is the largest organelle in a eukaryotic cell. It's often called the control center because of the fact that it holds our DNA in there. And why is DNA important? Well, DNA tells what proteins will be made and when they will be made. So it's the brains of the entire operation. So if you're thinking about a cell, let's talk about like a heart cell. You're going to need certain proteins for those types of cells compared to nerve cells. All proteins have different functions. There are so many proteins out there that do different jobs and some that do multiple jobs. But heart cells are going to tell heart cells, their DNA is going to tell the cell, you need to make this type of protein so we can make sure that the heart is pumping continuously. In comparison to that, if we had like the nerve cells and they had DNA in them, same DNA, but now we're telling this nerve cell, you need to make this protein so that we can send out stimulus throughout the entire body. So all of these have functions, but they may be just a little bit different. Doing the same job, just a little bit different. Next up, we have what's known as the mitochondria. Never will you call it the powerhouse of the cell. I absolutely hate that definition. I know why it's called that. I just don't like it. What the mitochondria does, and the mitochondria is actually the plural version of it. When we're talking about just a single mitochondrion, or like one of them, it's called a mitochondrion. And this is an organelle that makes energy available to the cell. So basically, when we eat, we take in the glucose, that's the important sugar in all foods. We take that in, we break it down, and we make that glucose into what's known as ATP. And ATP is like the currency of the cell. It is the money. It's like if I go to the casino and I'm I shouldn't say the casino, any kind of place, and I need tokens for them. So I'm going to use my dollar. I'm going to put in $1 into the machine, and it's going to give me tokens for these games. I don't know if you guys know what I'm talking about, but these tokens are used as currency for the games, and that's how it works for us. We input glucose. Our cells don't use glucose because it's too big for them, so they change it into what's known as ATP, and that's energy for the cell. And believe it or not, cells actually need energy in order to do their functions. Everything in our body requires energy. So the reason why you feel so sluggish and tired if you do not eat a meal or you skip meals is because you're lacking in glucose. Therefore, your energy level is decreasing and decreasing. The idea of being hangry is a real thing. When we talk about the mitochondria, one special thing about it is that it has its own set of DNA. Very special. And we actually get this DNA from our moms. So shout out to moms. But there's a theory behind that. It is called the endosymbiotic theory. The idea that a small prokaryote was eaten or engulfed by a larger prokaryote. And it didn't seem to hurt either one of them. They worked very nicely in this symbiotic relationship, so much so that the big prokaryote was getting extra energy from the smaller prokaryote, and then the small prokaryote had a place to live. Eventually, this probably became a permanent thing or a permanent guest in the cell, and then this was passed on. And again, this is a theory, so there may be another answer, but that's the most widely accepted theory of how the mitochondria has its own DNA. Endoplasmic reticulum. There are two of those. So basically what the endoplasmic reticulum is it makes and transports proteins and lipids. And what's so special about these proteins and lipids, not proteins and lipids, but about the endoplasmic reticulum is that there are two of them. One is called the rough endoplasmic reticulum. The other is called the smooth endoplasmic reticulum. You can just go ahead and refer to them as the rough ER and the smooth ER. The way you can tell the difference in what they look like is the rough is actually studded by ribosomes. So it's like embedded in them. And those ribosomes, obviously, that's where proteins are made. Hopefully we remember that. If you don't, that's just fine. But the rough is studded, so it looks like a sheet with like dots all over it. Whereas the smooth is not studded, does not have ribosomes, but the smooth is used for like making lipids, storing other features, but mostly making lipids. The rough endoplasmic reticulum is responsible for not only holding the proteins or housing the ribosomes, but vesicles and vacuoles. And what those are like little pouches that go and send proteins on their way. After that, we have ribosomes, and ribosomes are where proteins are made. That's a pretty big job, pretty significant. I'm not going to go into how proteins are made just yet. We will talk about that in a whole chapter. That's a whole different thing. But basically, you just need to know where proteins are made. And remember, proteins are the workers of the body, so super significant and super important. The Golgi apparatus, also known as the post office of the cell. So once it picks up proteins that are shipped off from the ribosomes that are on the rough endoplasmic reticulum, they will receive them, package and label them, and then send them out to their destination. So different parts of the cell, outside of the cell, all of these things are processed through the Golgi apparatus, and it sends them on their way where they need to go. Vesicles and vacuoles. I kind of already spoke up on these, but these are sac-like organelles that store and transport material in the cells. They don't only store and transport proteins. They can store and transport a number of things. There are special types of vesicles known as lysosomes and peroxisomes. You don't need to know what those do. I think in my notes I talk about them just a little bit, but not a whole bunch. You don't need to be specific, but just know that vesicles and vacuoles are really used for transporting. So if the Golgi apparatus is the post office, then the vesicles and the vacuoles are like the truck that gets them from point A to point B. And then the next thing we have is the centrioles. I cannot talk today. And centrioles are really, really special because they're only actually found in animal cells. And they are involved with cell division. So when we go from one cell to two daughter cells, that the centrioles play a huge significant role in that. That's really all I want you to know for now when it comes to centrioles, that they're only found in animal cells and that they're involved in cell division. Last but certainly not least is the idea of plant cells. Now, plant cells are a little bit different than animal cells. So far, I have described eukaryotic cells in general. When we start talking about the difference between animal cells and plant cells, there are a couple distinguishing marks. Plant cells are actually have a little bit more of a protective feature. Remember where plants live. They typically live outside. They're exposed to a lot more. They don't always have this nice like as humans, we have this thick skin and we have a lot more protective measures in that way. So plant cells have evolved to create a lot more protective features. One of those things is what's known as the cell wall. So a nice rigid layer that surrounds the plasma membrane. So we have the plasma membrane and then the cell wall, and that is the outside of the plant. So it protects and supports the cell, allowing it to grow up. And it's made of a really nice hard substance known as cellulose. In a plant cell, there you can also find the central vacuole. This vacuole can be made up of 90% of the cell's volume. So it's a really large. It really looks like a big pocket inside of the cell. It stores substances like water, enzymes, salts, all of these things that the plant cell may need in time of distress. So it'll pull from the central vacuole. It also helps plant tissues to help them hold their shape and stay rigid. It even gives flowers beautiful colors. So that's pretty fun for the large central vacuole. And last but certainly not least, we have the plastids. Plastids, what those are various organelles in plant cells that carry out different functions. And I've given you like 3 examples. So chloroplast, chromoplast, and leucoplast. They have different jobs. So chloroplasts are responsible for green pigment, such as chlorophyll, which helps capture light for photosynthesis. Chromoplasts give other pigments such as red for flowers and plants and various things like that. Or not plants, leaves. And then, leucoplasts are plastids that store substances such as starch or make small molecules such as amino acids. I really just want you to know that plastids, and there are 3 different types. Don't freak out if you're like super confused. One thing you should know though is that plastids contain their own DNA. So really, really special, just like the mitochondria in all eukaryotic cells, plastids also have their own DNA. So similar theory of the endosymbiotic theory, but a little different because now we're talking about plastids. All right, that is your video. I truly hope that this isn't super long, but if it is, I'm kind of sorry. If you have any questions, please make sure to ask me on Edpuzzle. Also, if you are listening to this, I will give you 1 point of extra credit if you put a smiley face in for your question answer. This will show me who's actually listening and who is not. So a smiley face for the question, 1 point of extra credit. I'm totally kidding. I'm not giving extra credit, but go ahead and put the smiley face anyway. Have a great day.